I had mentioned in the earlier session, there's a process called coagulation. In fact, coagulation is part of hemostasis. Whenever there is a rupture in the blood vessel or in the tissues adjacent to it, or maybe a major injury, the first thing that happens is hemostasis. Hem stands for blood and stasis stands for halting, stopping. So blood flow or loss of blood through that ruptured tissue needs to be stopped, needs to be halted. That is called as hemostasis. It involves three main stages. The first stage is vascular spasm. That is, the blood vessels going towards the tissue where there is a damage, they start contracting, they start constricting. There's a process of vasoconstriction which is initiated so that the blood flowing towards the damaged tissue is either reduced or stopped. So vasoconstriction or vascular spasm, as I said, is the first part of the process. Second part of the process would be formation of platelet plugs. Platelet plug is number of platelets come to the site of the damaged tissue. They start aggregating and they kind of start forming a plug that blocks the damage in the blood vessel. This platelet plug formation, we will talk about that process in details, is the second process. And the third process that follows it, it's the coagulation. Coagulation cascade, as I had mentioned in the earlier session, that coagulation cascade is a series of reactions. It's a flow of processes that finally ends into reducing the blood loss or minimizing the blood loss or maybe stopping it completely through the wound. Okay, let me explain the process of platelet plug formation. Now here, whenever there is a damage in the blood vessel, the first tissue damaged happens to be the endothelial lining, innermost covering of blood vessel. But then platelets do not have any attraction towards endothelium. So endothelial cells, endothelial tissue, endothelial lining doesn't attract the platelets. But the tissue next to endothelium, that is the connective tissue, when it is damaged, the collagen fibers which are part of the connective tissue, they get exposed. And it is the collagen fibers which attract the platelets. So platelets start moving towards the collagen fibers. At the same time, when collagen fiber is attracting the platelets, endothelial cells, they start releasing a chemical which is called as von Willebrand factor. Von Willebrand factor is something which enhances the reply by platelets. More and more platelets start moving towards the collagen fibers. Now, as these platelets start moving towards the damaged tissue, they start moving towards each other. And for staying together, they undergo certain changes in the shapes or the morphology. They start swelling up, they start showing certain protoplasmic extensions, spike-like extensions they form, they become sticky and they form a kind of aggregation. At the same time, they start releasing certain chemicals. The first one is serotonin. Serotonin helps in enhancing the vascular spasms, the vasoconstriction process. Another chemical which they release is ADP, adenosine diphosphate. Now, ADP increases the aggregation of platelets. There's a release of a large protein, a kind of prostaglandin, that's called as thromboxane. Thromboxane stimulates the release of serotonin and the ADP by platelets. At the same time, the endothelial cells, they start releasing another chemical that is called as prostacyclin. It's another kind of prostaglandin. Now this prostacyclin in fact avoids the aggregation of platelets. It doesn't let the platelets stick together. So the resultant platelet plug, which is formed to cover up that damage in the endothelial lining, it's a loosely formed congregation of cells. The cells are not tightly packed together. So platelet plug actually cannot completely avoid the loss of blood. But simultaneously, there is triggering of coagulation process that results into formation of certain protein fibers, polymers called fibrin fibers, which can bind the platelets together and form a platelet plug, a tightly formed platelet plug that would prevent further loss of blood through the damaged tissue. Okay, let us try to understand the coagulation cascade process. Exactly how does it happen and what all factors are involved. Now this process involves a large number of coagulation factors. Almost 13 different factors are involved in the process. As I mentioned earlier, the process begins when a blood vessel or a tissue is damaged. That ruptured blood vessel, the innermost covering, as we have seen, it's endothelium. Next to that, it's a connective tissue. Now endothelium, as I said, it actually doesn't invite any response from the platelets, but the connective tissue where it is damaged, the collagen fibers which are exposed, they start attracting the platelets. So platelets start moving towards the collagen fibers from the connective tissue. Now as the platelets start moving in, a number of factors, coagulation factors contained in the plasma, they start participating in the process. The first factor is factor 12, which is called as Haemostasis factor or glass factor. That is the first factor to be activated in the process. It is activated by the aggregating platelets. That factor 12 or Haemostasis factor, after activation, it goes and triggers the activation of factor 11. Factor 11, which is called as plasma thromboplastic antecedent, PTA. PTA is activated by activated factor 12. So factor 11 becomes active now. This activated factor 11 then goes and triggers the activation of factor 9. Factor 9 is called as plasma thromboplastic component. It's also called as Crispus factor. This Crispus factor is activated by factor 11. Now this activation of Crispus factor, it also requires presence of calcium ions and factor 7. Factor 7 is also called as proconvertin. It's also called as SPCA. Proconverting or SPCA, it activates factor 9. Now factor 9 would then go and activate factor 10. Factor 10, which is also called as Stewart-Prower factor. Stewart-Prower factor. This activation of Stewart factor, it also requires presence of platelet factor. Platelet factor, that is platelet factor 3 it is called, which is released by platelets. At the same time, it also requires presence of factor 8, that is anti-hemophilic factor, AHF. That is also required in activation of factor 10, Stewart-Prower factor. Once the Stewart-Power factor is activated, it goes and triggers the activation of prothrombin activator. Prothrombin activator. Now this activation of prothrombin activator requires again presence of calcium ions and platelet factor 3, and at the same time it requires presence of activated factor 5, that is proaxelerin it is called as, or it's also called as labile factor or platelet accelerator. That requires to be present for activation of prothrombin activator. Prothrombin activator then converts inactive prothrombin into active thrombin. So prothrombin is converted into thrombin by prothrombin activator. That thrombin then triggers conversion of fibrinogen, an inactive component, into fibrin, an active protein. Thus again, conversion of fibrinogen into fibrin requires presence of calcium ions and factor 13. Factor 13 is fibrin stabilizing factor, FSF, fibrin stabilizing factor. Now, once fibrin is activated, once fibrinogen is converted into fibrin, fibrin starts forming cross-linked polymer, cross-linked fibers. A large number of these fibers, they start forming a network, a mesh, into which the platelets and the worn out red blood cells start getting trapped, and that leads to formation of a blood clot. See, platelet plug is almost colorless or yellowish in appearance. But this clot that we are talking about, because the red blood cells are being trapped in it, it becomes more or less dark red or brownish in color. Once this is formed, once the blood clot is formed, that would seal the damage in the blood vessel and further loss of blood will be stopped. That means, as I said in the earlier session, earlier part of the session, hemostasis helps in stopping the blood loss. This is what exactly is happening here. At the end of this coagulation cascade, there is formation of a blood clot that would seal the damage in the blood vessel, and further loss of blood through that place would be checked, would be prevented. In the extrinsic process, wherein we are talking about damage to a tissue from outside, external tissue, this is similar to the earlier process which I discussed from a particular stage. Initial stages would slightly differ. The first thing that would happen is, whenever there is a damage in the tissue, the damaged tissue or cells from the damaged tissue, they start releasing a tissue factor, TF. This tissue factor, first of all, participates in activation of proconvertin. That is what I earlier referred to as factor 8. 7. This proconvertin would then help in activation of factor 9. That is, in presence of calcium ions, proconverting will activate factor 9, that is plasma thromboplastic component or Crispus factor. Rest of the process would remain the same as I discussed in the intrinsic coagulation. Now, once the plug is formed, once the clot is formed and the damaged blood vessel is sealed completely, it would first of all avoid the further loss of blood. Secondly, the white blood cells in the vicinity, they start releasing a secretion that becomes a serum. It forms a thin layer over that clot, and this deposition, this secretion would prevent infection of the wound. Bacterial or fungal growth would be checked by it. And the fibrin fibers, gradually they undergo shrinking. As they dry, they shrink, and thus the two edges of the tissue, they are pulled closer, and that helps in healing of the wound. So clotting process would have three advantages. First of all, it would prevent loss of blood. Secondly, it will avoid infection of the wound, and thirdly, it will help in healing of the wound. That is what the coagulation process is. Keep watching the videos, do like them, subscribe to them, and share them amongst your friends and family. Thank you.